afternoon to all of you. Today we are back after the sessions and we'll concentrate today on pure culture technique. So, pure culture is a most important aspect regarding microbiology because in order to analyze any sort of sample which comes in the microbiology laboratory, we need to compare that sample with the standard samples or with the standard microorganisms that are present. So, in order to isolate the organisms from the group of organisms, we need such kind of techniques which are so called as pure culture techniques. So, let us understand that a microbiological culture or a microbial culture is a method of multiplying microbial organisms by letting them reproduce in predetermined culture media under controlled laboratory conditions. Means microbiological culture or microbiology is the method of microbial organism by letting them reproduce. See, when the organisms are coming or when the organisms are there in the sample, we need to isolate the organisms in a higher amount. Because for example, if we isolate a single organisms and by mistake if we lose that organism, then it is absolutely of no use to us. So therefore, in order to increase the quantity of the organisms, but in a pure form also, we need to work out with this kind of methods. Microbial cultures are used to determine the type of organisms, its abundance in the sample being tested or both. It is one of the primary diagnostic method of microbiology and it uses a tool to determine the cause of infection by letting the agent multiply in predetermined media. So therefore, in order to identify that what is the causative part from the sample, for example, if a particular patient of tuberculosis comes and he gives a sputum sample, then inside the sputum there may be many organisms, but what we should concentrate on only for tuberculosis. So what we'll do, that we will try to isolate the organism only for tuberculosis. For that purpose, we need a pure culture to be getting isolated, and therefore these kind of methods are said to be helpful. So microbial cultures are a foundational and basic diagnostic method, and for in terms of molecular biology, isolation of pure culture method. A pure axenic culture is a population of cells or multicellular organisms growing in the absence of other species or types. Pure culture, yani kya? What we can define a pure culture to be? Pure culture is something which is free of other organisms. Means, for example, if a sample, just now what I told you in terms of tuberculosis, in a similar way, when any sort of food, for example, if you bring any sort of food from the market and if you try to isolate organism from it, there may be number of organisms inside that particular food particle. But if we want to concentrate on a single organism or a single bacteria from the rest of the population, we need to separate them, and that separation is called as purity of the particular sample. That purity or the pure culture is also called as axenic culture. This is most important term. It may be asked inside the examination as an MCQ as well. What is pure culture called as? Therefore, it is also called as axenic culture. Population of cells and multiple organisms in the absence of species other than. Pure culture may originate from a single cell or single organism in which the keys in the cells are genetically clones to one. Means from a single cell itself also we can be able to isolate the pure culture. See this. Introduce bacteria into the growth medium under aseptic conditions to prevent contaminations. What we need? We need the Bunsen burner, loops, needles, etc. See this, nichrome vial. Very small in size and we need burners. Why burners are essential? In order to maintain the aseptic condition in that particular area, we need the burners to be there. See here, this person is transferring with the help of a nichrome vial loop, it is transferring the viable organisms into the fresh medium. We have already discussed the part in terms of media. Media may be either solid media or maybe either liquid media. Now, one of the basic methods that is observed in case of microbiology is called as pure culture technique or pour plate technique. So this pour plate technique, how one can do it? Pour plate technique. This is the medium and inside this particular medium itself, the neck of the agar bottle is pressed to the flame. It has to be passed under the flame. Question arises, why to pass under the flame? The reason behind it is that because of this flame, direct flaming, the microorganisms which may be present or which may have contaminated this area may be removed because of the direct burning method, and therefore it has to be passed from the flame so that the excess amount of bacteria that are present over there will be died, and you get the pure form here. There will be no contamination inside here. Slowly it has to be poured. See the way it has to be poured. And see, every time it has to be under sterile condition only. Very good. So once this has been done, you have to just cover that particular plate or allow this plate to be stable for a long period of time so that the media gets cooled down. Once the media gets cooled down, because of the presence of the agar inside the nutrient agar and any sort of medium, the plate slowly gets solidified, and ultimately the sterile molten plate can be developed. But all these conditions has to be maintained very correctly inside the Bunsen burner only, so that we will be able to get pure form of culture to be isolated. Okay. So there should not be any prior contamination before we start our experimentation. Therefore, this particular kind of technique can be done to prepare a plate, microbial plate, okay, for medium. Aseptic technique. In working with microorganisms, we need to have a method of transferring growth organism from pure culture to sterile medium without introducing any unwanted outside contaminants. Now, for example, if we have a pure culture along with us and we want to transfer this pure culture to another medium, let us say. Now, what is happening is that we know that there are no other such organisms which are present inside this plate, and now we need to transfer it to another another medium. While transferring, if aseptic conditions have not been maintained, then it may happen that along with this, there are organisms in the air, there are organisms on that plate also, which may get contaminated, and your results may have been very good. This method of preventing unwanted microorganisms from gaining access is termed as aseptic technique. What we say as aseptic technique. For the most part, bacterial physiology only can be studied in pure culture. Now, in order to study the physiology of the microorganism, that what this microorganism is, its size, shape, morphology, plus what type of enzyme it produces, what type of fermentation it can work out. If we want to study all those things, we need to work in a very correct manner in order to isolate the pure culture. The best way to obtain pure culture is to start a single bacterial cell. This cell then divides quickly, may produce millions of cells within 24 hours. So, a very small amount of bacterial cell has to be cultured from or can be taken, and it has then to be transferred into another medium so that it will work out very correct. So, for that purpose, what we need? We need the nichrome wire loop. What is the nichrome wire? An alloy of nickel and chromium. Now, question arises many times. Students always ask that why this nichrome wire loop cannot be of iron. This nichrome wire loop has an ability. It may be of platinum wire also. These metals, which are alloy of nickel and chromium or platinum, have an ability is that once you put them inside the flame, they get hot very early. Correct? And when you put them outside the flame, after they getting red hot, then if you put it outside the plate, then certainly it will be more better for us or more better to cool down very fast. So this metal has both the ability that it gets hot very high at high temperatures and it cools down very fast as well. Therefore, basically this can be used. If you use iron instead of nickel or chromium, then iron, if the iron gets hot, if red hot, if we go along with doing red out to that particular iron, it will take at least 3 to 4 hours to cool down. So that experimentation cannot be worked out. Therefore, we use generally the platinum wire or nickel and chromium alloy wire. Now, the similar instrument in inoculating needle is actually the same as well. Sterilize both the instrument by holding the wire portion. Here we have to there. Now, before that, contaminants of the wire can be eaten. Do not blow on the instruments to cool them. This cannot be done. Why is it so? Because there are bacteria that are present inside your mouth, and again you are contaminating it onto that particular nichrome wire. Do not touch the instruments to agar to cool them. Never. If they are so much hot and if you put it onto the agar, then it will go down directly. It will just liquefy the agar as well, because agar, you know, agar once a little bit amount of heat is being provided, that agar will again melt. So that cannot be done. Do not lay the loop down once it is sterilized, and it may become contaminated. Once it has been sterilized into that particular burner, you cannot put that down. You have to perform the experiment and then you have to put down. Okay? See this. This is the inner core, interpole gases and outer diffusion vectors or outer core of the burner. This has to be maintained. The blue flame has to also be there. Now, how transfer takes place? Flame the loop. Without setting the loop down, open the first culture tube and flame the mount. Do not set the cap on the bench, and the cap should be held in the mount. Insert the loop inside the culture medium and then withdraw. Flame the mouth of the culture tube again and replace the cap. Now, this is most important. Indication of culture. To isolate bacteria in the pure culture, what is the need of this aseptic technique that has to be mentioned? Isolation of bacteria in pure culture. Demonstrate their properties, because once we get a pure culture, then and only then we will be able to demonstrate the properties. Obtain sufficient growth for the preparation of agents and for other tests. So because once, if you have a sample in a very small amount, then it have a maximum chance you lose the sample very well. Therefore, in order to, if you don't want to lose the sample, then the sample can be generated with a very small amount. Many number of bacteria can be generated, which will be similar to its original counterpart. Typing bacteria and isolate means what sort of bacteria are present over there that can be easily clarified. Antibiotic sensitivity. Now you need to have a particular kind of what we can say pure culture. Then and only then you can easily detect the resistance of the particular bacteria. Estimate a viable count. How many number of bacteria are cultured inside that medium can easily be demonstrated. Maintain stock culture. In order to maintain the stock cultures, this can be easily be viable. Types of culture. 3 different types of culture methods can be utilized: stick plate, pour plate, and spread plate technique. Stick plate. See this. In case of stick plate technique, you need to go along with this. If you come to this part, spread the knuckle back and forth and across 4 quadrant. This is the 4 quadrant method. See this. Quadrant number 1. You start your loop. You take organisms inside this loop and you start sticking like this. This is the first stick. Then you move the plate with your hands itself, but this has to be done inside the burner only. Then this is the second quadrant. Easily we can do along with this. Again, just tilt the plate. This is the third quadrant. Again, the plate has to be done, and then this is the fourth quadrant. Now, once you incubate this, after this you have to incubate inside the incubator, and after incubation, you will observe that on the first stick, here there will be load of organisms will be very high, because we are putting directly from the microbial loop, we are putting it onto that particular media. So the rate of microorganisms or the density of the microorganisms will be very high. Now once you come to this part, a little bit dilution can be seen over here. So this, what we can say, the concentrated part is now being diluted a little bit. Again, if you have turned the blade with this method, from here you are again diluting it, and you can easily observe the bacterial colonies over here. After this, with the green portion you can see over here that we are diluting this part also, and well isolated colonies can be obtained in the fourth quadrant. So this is how the stick plate method works. Why it is called a stick? Because these are sticks are lines that can be drawn onto that particular plate, and you can easily get a well isolated colonies over here. Correct. Second type of method that has been developed for this particular kind, it is called as the pour plate technique. For this pour plate technique, 1 ml of appropriately diluted inoculum is added to 15 ml of molten agar and poured on the petri dish. Now this is the molten agar, but this contains, what it contains? It contains dilution. So after diluting it, see this, 10⁻² dilution, -2 dilution, and diluting that culture may help you to get well isolated colonies onto that agar. What we have to do inside after dilution, you can pour it the molten agar, or you can pour it into that particular tubes of molten agar, mix it properly and just pour it onto a particular kind of sterilized plates. Then after that, based on the dilution itself, you can easily get observe the isolated, well isolated colonies. Spread technique. This is the third technique that can be mentioned. Prepare a layer bench with required material, dilute the sample and shape gently to suspend organism. Insert the loop, insert the test tube and remove it with bacterial broth. See this, I will come to this point so that it will be more clearer. See this is the pour plate technique, very correctly, you can easily see over here. Now, spread plate technique. In case of spread plate techniques, the sample can be taken and it has to be poured onto the solid medium only, and with the help of a sterilized spreader, you can spread it in all possible direction by just tilting the plate. You can easily spread it in a very unique manner. After incubation, you will get the well isolated colonies can be seen. So these three techniques are said to be one of the most important techniques in case of isolation of pure culture. So if you want to isolate the pure culture, just go along with these kind of methods and easily one can get that particular kind of thing. See this. After see the drawings that have been made so nicely. See the stick plate method. This is by pour plate. See the different types of bacteria you can easily observe. This is the media that can be here, and after the media has been used with the help of organisms, you can see the number of organisms that can be easily be seen in here. Okay? So we'll stop it for today, and what we have learned three different techniques. One is stick plate, pour plate and spread plate technique for isolation of pure culture. Okay.